Okay, so this video actually gets to the core of statistical design principles. We talk about independence, randomization, replication, and representation of populations by samples. So this video will be a little bit longer and more complex. I actually have to start by explaining how statistics work in principle. But it doesn't make a lot of sense to break it up, so this all builds on each other and is all related to each other. Okay, so let's talk about independence. That's actually an important concept, and I am going to mark those videos that convey these important concepts with a little star in the thumbnail as well, so that you can review them selectively if you want to. So those are the videos that you don't want to miss. So you may have heard of independence as a statistical assumption. If you ask anybody, what are the assumptions for any random statistical test? If you say normality, homogeneity of variances, and independence, you can't go wrong. But the order actually should be different. So if you start with the most important, start with independence. So independence is really an important assumption for any statistical test. Homogeneity of variances I would rank second. And then normality really is not that important. You can either ignore it or you can find a test that doesn't have the assumption of normality, so that's really not a big problem. But if you violate the assumption of independence, that's a showstopper, so you're done. So there's nothing you can do. Your whole study is for naught. So in the applied sciences, there's lots of examples where you can't be quite sure, but you still have to make decisions. Management decisions, public health decisions, business decisions, you name it. So if you're making statements about the world, statistics can tell you how likely it is that you're actually right about that. So I've drawn this little example here for such an applied question in forestry. So if you're a forester operating in a particular region, you might ask the question, is it better to plant lodgepole pine or white spruce in a particular ecoregion? That's my population. That I want to draw inferences on. And generally the population is not necessarily all trees in the world or all individuals in the world. Typically the questions you're asking are slightly more narrowly defined. So in our case, let's say the population is the foothills forest region of Alberta, and I'm interested whether lodgepole pine or white spruce have better growth rates, better wood quality, and I want to make a decision as a forestry company which trees to plant. Now, one thing I could do is, if I really want the truth, I could measure every single tree, and well, that's about I don't know, maybe 10 billion trees. So if you measure them all and you come up with a result that says lodgepole pine is 17.58796537.2% better in growth rate than white spruce, sure, you're done. That's the truth. There's still room for measurement error, but my guess is you could get a paper out of that if you really did that study. So in that case, you actually don't need any statistics. And while this might sound like a rather implausible example, there are plenty of studies where you actually do work with a population. So whenever you study a population that's relatively small, of high interest, and you keep track of all individuals, you don't need statistics. So this could be a conservation application where you work with an endangered population in a particular area. So if that's your focus and you keep track of all the animals, there is no need for statistics. And those can also be global studies. For example, if you do remote sensing, most people don't realize it, but it doesn't make a lot of sense to do classical statistical tests on remote sensing data where you have wall-to-wall coverage. You work with the population, so there is no need for significance testing or things like that. You just describe how things are based on a complete set of information. But in our case, the statistics will be very useful. So instead of measuring 10 billion trees in this area, maybe I just get away with 10,000 trees. So I won't be able to say lodgepole pine grows 17.6798 and so on % better than white spruce. But I could potentially make a statement that lodgepole pine grows at least 15% faster with a 98% probability. And that 98% probability reflects a p-value of 0.02 in this particular example. Now, if I only sampled 100 trees, I'm naturally less certain about the results, so my result that I report back, maybe I'm only 70% certain that lodgepole pine grows at least 15% faster than white spruce. But this is still information a decision maker can work with, even though in this case I only got a p-value of 0.3. So p-values don't actually need to be high to inform decisions. Our pandemic is another great example for this. So public health officials, they can make excellent policy recommendations based on scientific advice, as long as that advice comes with a statement of uncertainty as well. So if you don't like my forestry examples, keep in mind that whatever I explain here works for every field of science. So if the question the question that I want to answer is, should I roll out a vaccine to the general population? My population is, in this case, all individuals in the world, humans, and my sample, instead of comparing white spruce and lodgepole pine, I would compare people vaccinated with a candidate vaccine versus a control group that got vaccinated with a placebo. And the question I would be asking would be exactly the same type, right? So the statements that I want to make may be, I want to be 80% sure that that vaccine reduces my infection rate relative to the control group by at least 30%. But then I also want to be 99.8% sure that nobody dies from side effects. So there are different tests with different p-value levels that I need to carry out, but the principles are always the same across any field of science. So one thing that determines the uncertainty is the number of samples I take here. So that certainly has a big impact on the certainty with which I can make a particular statement. But the quality of samples is also important. Now, before we get there, I do want to wrap this section up with some definitions and some statistical vocabulary. So we mentioned populations quite a bit. So those are the entirety of objects that you want to make a statement about. That's not necessarily in every individual in the world, so it can be a much narrower scope. Samples are a subject of objects that you actually study. So those are the ones that you measure, those are the ones that you investigate. And the way statistics works in general is that we use samples to draw inferences on populations using statistics. So in statistics, narrowly defined, are actually property of a sample. So it could be the mean of a sample, and what we infer is a population parameter. So we infer that my mean growth rates for spruce versus pine are particular values. Okay, so this gives you an idea how statistics works in principle, and this is really the whole story. This is what statistics does. So now you know. So let's proceed to talk about independence of samples. And one thing that ensures independence of samples is randomization. But there's actually more to the quality of samples than just randomization. It's not a black and white attribute of samples, either randomized or not. There are other more subtle considerations that also depend on the context, that depend on your question. I like to use the analogy of a court case actually to explain what makes a good sample. So you can think as yourself as a claimant, and each row in your data table would be one of your witnesses who testifies in support of your claim. And we already established the more witnesses you have, or the more samples you have, the more certain you can be of your statement or of your claim to be successful. So that definitely works in statistics as much in a court case. If you have an impressive lineup of hopefully completely independent witnesses who corroborate your story, you have a strong case. But it also depends on your claim. If we take the example that we had in our data table, let's say we're testing a new variety on a number of experimental farms. If your claim is that farmers across Western Canada should plant variety B in the future, then you would want a sampling design like this. So if your sampling design looks like that, that would raise an objection. This is speculation, conjecture. So your witnesses are actually don't know what they're talking about. They're just voicing an opinion on incomplete information. So representation relative to your statements, those two things have to match up. So in science, you generally want to draw broad inferences, make your sampling design as large as possible, and sometimes when you interpret your results, you get carried away and you make claims that are really not quite supported by your data. And that's not necessarily a deal breaker. You can always climb back and narrow your claims, but you also have to be careful on how far you actually have to climb back. So at some point, your case may be dismissed because it's not in the public interest. So if you have a study that is so narrow that it's really only of interest to a very small audience, for example, we see that quite a bit in our research on land reclamation. There are very specialized studies that are just applicable to one quarry of one company in one location. And it's difficult to actually make any broader inferences for anybody else. So that has to come in at the design phase of your study, that you're not working on too narrow a field. So the next objection you may encounter is hearsay. So that's at the heart of independence. And as you can see here, what you need to do to have each sample be an independent witness in support of your claim, those samples need to be randomized. So if you have your samples for treatment 1 here, let's say that is a ecological study of 3 successional phases, and you have a forestry strand over here that's early successional, mid-successional, and late successional. Any difference that you observe, they could very well be due to different successional stages, but it could also be due to any other factor. So it could be that this area just has different soils, or this area is better drained, or if you have a big spruce seed tree here, those might not be spruce forests because they are late successional, because they had a big spruce tree nearby that seeded that area. So this is a weakness. This is called a pseudo-replication. So if you don't have randomized data, that is a problem. And you can see that randomization would really be effective, because if I had high spruce occurrence in this field, and this field independently, that's a much stronger case. So randomization, both in sampling designs and experimental designs, is essential. And oftentimes it's actually not easy to lay out a treatment. For example, if you have agricultural treatments that require a big tractor plowing across a field, then you often get these entire rows, and you may be tempted to put your replications into these rows of different treatments. But these are not independent witnesses. So they are all talking about the same thing. They've been all talking to each other. They've all been subject to the same conditions. They are not as credible as independent witnesses over here. But many times you're actually trying to strike a balance between randomization and logistics. For example, you might have forestry plots with different harvesting treatments, and you want to understand the effect those treatments have on your insect population. So you might, within those plots, place transects that then have insect traps. One advantage of this is that you have good representation of your larger plot. So if you only put one insect trap in there, that is not very representative of that treatment. But ultimately, you need also replications of your main treatment level that you want to make statements about. So if a partial harvest is better than a clear cut or something like that. So those compromises are fine, but you have to be a little bit careful, because if you put the results from those traps or from those transects into rows in your data table, you do violate the assumption of independence. So your treatments are independent, A and B. So those are independent replications, and that's good. But within your treatment plots, whatever you collect there, this is spatially autocorrelated. So these are not independent. And so you either have to summarize them, or there are statistical methods that account for those spatial autocorrelations, which we may get to later. Those are more advanced analytical methods. So restricted randomization, that is actually fine, as long as you have at least one true independent replication. Then you're in business. So if you're in this situation here where none of your replications is a truly independent witness, then you're done. So those studies do not go anywhere. They don't make sense. There is just too much uncertainty. You can't disentangle environmental gradients that you may have in your study area. So this is a real problem. And if I give an analogy here for this situation in a court case, this is really akin to my word against your word. It could be the treatment that causes the effect, or it could be something else. Nobody knows that. And unfortunately, these studies are quite common. So a lot of junior students get put on pilot projects, and there is no money for a big project with big replications. But if you find yourself in a situation where somebody asks you to, let's say, compare a polluted lake versus a control lake and put lots of work into to assess water quality and riparian vegetation, for example, try to make a case for at least one replication. Another example are greenhouse studies or growth chamber studies. Growth chambers are expensive, so tying up a lot of growth chambers for replications, that's usually out of the question. But if your supervisor puts you on a project like this, or if you find yourself in a job where that sort of project is proposed, you don't need a lot of replications, but one replication. One replication makes a world of a difference. And you can see why. So if you have a court case where it's your words against my word, so you say this, your opponent says that, the judge throws the hands in the air and says, well, I don't really know. There's nothing here I can base a judgment on. But if you have one independent witness who corroborates your story or the story of your opponent, it's a clear result. You win the case or your opponent wins the case. So having that one replication does make a world of a difference in science. And just as an interesting aside, many people struggle with what degree of freedom means in statistics. So this is a really hard concept. But what it really means is the number of truly independent witnesses. So those are the number of samples that are free to vary. And this analogy interestingly holds even further. So if you have a very complex experiment, you need a lot of degrees of freedom. And similarly, in a complex court case with many aspects, you would need a lot of independent witnesses to make your case. But you can make a mental note that this is what this is all about. So the number of truly independent witnesses, that's your degrees of freedom in a statistical design. Okay, to finish this video off, I want to push my court analogy even a little bit further to give you some hints on what kind of research or projects you might want to engage in. So this is my last point here. If you make frivolous or trivial claims, your case may be dismissed. So the nature of your claim, the nature of your statement is actually the thing that matters the most. And, statistics can help you to make a judgment given the evidence presented. Are you right about something or are you not? Or is it likely that you're right about something? So a judge can do this with lots of evidence presented before him or her. And statistics can do that too. But the question is, should your case be heard in the first place? So the analogy in science is that your paper gets rejected before review because it's simply trivial or a frivolous claim that you're putting out there. So showstoppers are clearly if you don't have any independent witnesses, but also if you don't have a question that makes a lot of sense, that doesn't have applied value, that is not of scientific interest. So the example I gave you, that certainly had applied value in the very beginning. If you remember the lodgepole pine versus white spruce experiment, made the statement that lodgepole pine grows at least 15% faster than white spruce, and our p-value was 0.02, which implies a certainty of 98% that you're actually right about that. But you could screw this up quite easily And so one thing that is actually not so bad, so I highlighted that here in orange, if you can only make a claim if your sample size is not very large, and you can only make a claim that lodgepole pine grows 15% faster than white spruce with P 0.2, that may be non-significant, but it also still means that you're 80% sure that you're right about that. And a forestry company would be very pleased with such a result. They can act on that. There's no issues. And you could even make a good case in a journal that this is worth publishing. So don't be discouraged by p-values 0.05. They really don't mean anything. This is just a convention. It depends on the context. It depends on the statement you're making. Now, on the other hand, if your claim that you put out there is lodgepole pine grows faster than white spruce, and you get P equals 0.002%, so that seems like a high certainty, 99.8%. That sounds great, but it might actually be rejected as a trivial claim. So this one is not useful. It doesn't have any applied value, because by how much, If it only grows 0.1% faster, that means nothing from an applied perspective. So the magnitude of your differences, those are the ones that you actually have to report and test. So you can easily go wrong with your selection of the claim that you make, even though your statistics seem right. So keep that in mind when you design your own research or develop your own projects.